So good afternoon, everybody, and thanks, Eleonora, for the nice introduction. Today, I'm going to talk about new bioelectronic sensor for the early detection of protein marker, especially for the recognition of pancreatic-biliary cancer. At first, let's have a look at the problem here, and especially why we are so interested in this kind of biosensor and why on single molecule detection. What clinicians are demanding today is for a new sensor that can detect very low concentration of the analyte, and what is more, to discriminate between an healthy and a disease state in the very first stage of infection, in which we have for sure low concentration of the analyte, but we have no, the illness is not manifest. What we want to convey in this new device is to decrease the limit of the detection, so increasing the sensitivity of the sensor, and at the same time decreasing also the invasiveness of the sensor. So at least we can detect a small amount of the sample we collect, and for example, in blood or serum or saliva, so in a very non-invasive way. The problem here is that the most typical technique, like next generation sequencing or also the Quanterix SIMOA, are really nice also to detect genomic marker. But what is still unknown is the proteome, human proteome. In fact, if you look at this nice schema, the most abundant protein we have are the classical plasma proteins, such as albumins, but these are not bringing lots of information about illness state. Indeed, we have, for example, interleukins that have low concentration in the attomolar, femtomolar range that can be that can highlight a disease state, for example. The problem here is to focus on the protein also. Just to have an idea for why it is important to detect potential biomarker for pancreatic cancer, we can have a look to the path in which the detection is performed today. So at first, if we can see by imaging some lesion like tumor, we can directly go to chirurgical resection of this tumor. Or we can also have fine needle aspiration to collect the sample from pancreas, but for sure this kind of analysis is quite invasive. After this analysis, if we have a clear feature of malignity, then for sure we can go to removal, to chirurgical resection of this. But what if we have no clear feature of malignity? What to do? It is sick or not? Then here comes the problem and maybe the solution. So thanks to the pioneering work of Professor Torsi, we have developed a new bioelectronic sensor that can detect both genic marker like KRAS for the pancreatic cancer and also proteins like MAC1. And both these info are very important for clinicians to get info about the illness state. So in this way, we can detect a very small amount of this protein, for instance, even when there is no clear feature of malignity. So we can treat the patients early and have early diagnosis of the illness. Indeed, if low-grade lesions are there, the patients should be followed up just to decrease the probability that it could get this tumor. So this close the diagnosis circle and maybe give us a solution. As already introduced yesterday, we are working on this SYMBIC project that is basically focused on the single molecule technologies, in which we have an ELISA-like plate in which we have no optical readout, but bioelectronics. So on the bottom of this array, there are multiple transistors based on the FET technology, and the output we are collecting with this smart interface can give us info about the illness state by multiplexing, so we can get information of different biomarkers at the same time. So let's see how this looks like. We have a 96 ELISA well in which the incubation of the top gate electrode that has this 3D structure. So it is there is a gold on top. So this is the gate electrode. That is modified with the biorecognition element for the biomarker we are looking for example, the MAC1 protein. Then this is incubated in solution containing the analyte, is washed properly, and then is put in the reading system. So first of all, we have studied a single device, so not the array, but just one single transistor at time. And we have here a picture and a scheme of the device. So basically we have a source and drain interdigitated electrode on which the semiconductor is deposited by inkjet printing. Then we have the lateral gate on the bottom that serves as reference electrode. So it is a bare gold electrode that serves to prove that no variation is occurring on the semiconductor channel. And then on top, we have the 3D structure that is biomodified with the antibody, for example. So we put inside the well, and this is electrostatically connected through the electrolyte well with the channel. And we observe the variation on the transfer characteristic of the device. So by studying the relative variation of this current, we can discriminate between the presence of the analyte, for example, the MAC1 that is binding to the antibody. And then we have with a quite identical surface, so modified with BSA. In this case, we have no binding between the MAC1 and BSA. So we have this control experiment in which almost zero variation occurs. So then here I give you some nice picture with the whole device that we are working with, the single device. So this is a zoom on the lateral gate pad that we used as a reference electrode and the interdigitated source and drain electrode. And this is the 3D that how you can see here during the incubation, it perfectly matched the ELISA plate. What is important here, has already mentioned, is that the biomodified surface, that is a millimeter square area, should be fully covered of this biolayer. And so we studied independently a gold surface by means of multiparameter SPR. This gives us the opportunity to follow in real time any variation occurring on the surface. So here we can see all the steps of the biofunctionalization protocol during time. So at first, a self-assembled monolayer of alkyl thios is deposited on gold. Then we activated the carboxylic groups on top with the EDC NHS, and then the antibody is injected into the cell. So we perform here the covalent coupling of the antibody with the carboxylic group, and with SPR we can have also information about the surface coverage reached on the surface. That in this case is about 10 to the 11 molecule per centimeter square. So this means that we have a densely packed layer of antibody for catching the antigen. Then we use ethanolamine to saturate any possible carboxylic group that didn't react with the antibody. And then BSA is used here to fill any possible voids left on the surface and also to prevent a specific absorption. Also to verify that the antibody anchored to the surface is still active for the binding of the antigen, we proved also in SPR, for sure in the nanomolar range here, the binding at increasing concentration of the MAC1, and it was perfectly working. So we modified our gate electrode with the same procedure used in SPR, And we have performed the sensing. So the sensing gate modified with antibody that binds the MAC1, and the control experiment in which we have there only BSA. And here you can see also the trend basically in time. So the dynamics that we follow in the variation of the overall transfer characteristic. And so here I reported the maximum value of the drain current at the maximum VG, in which you can see the difference after the binding or at 100 zeptomolar concentration of the MAC1, in which we have almost a 30% variation compared to the initial baseline that was set in the reference serum. Then if you compare this with the control experiment, you can see that here we have an almost zero variation compared to the initial baseline. So basically, if you want more info about this, we also published during this year a paper on analytical and bioanalytical chemistry. Yeah, we have all the big family in SYMBIT, in which basically we are getting a world record in protein sensitivity at the single molecule level. And what is more, the system can both detect genomic marker and protein one. This could be super fast because the analysis can last even in 30 minutes. And there is also cost effective because you have only the disposable cartridge that is modified that can be replaced, so the transistor can be reused. And also we can have multiplexing platform there that can be also applied to different yieldness, for instance. For sure, if you want more info about SYMBIT, please visit our stand here in the conference. I want to thank all the group coordinated by Professor Torsi, and also the great work of Professor Gaetano Stamarcio, Dr. Paolo Bollella, and Dr. Eleonora Macchia, and all the partners of the SYMBIT project, and also the financial support for sure, and you for the kind attention.